In a diagnostic lab, the ABO and Rh blood grouping is done using a diagnostic kit that consists of monoclonal antibodies for A, B, and Rh antigens. Monoclonal antibodies against antigen A, also called antisera A, comes as a blue-colored liquid reagent in a small bottle with a dropper. Monoclonal antibodies against antigen B, also called antisera B, comes in yellow color, while the anti-Rhd monoclonal antibodies against Rhd antigens, also called antisera D, is a colorless reagent. All the color codes are universal standards. To perform the ABO and Rh blood grouping test, you will need the following: a clean glass slide or a white porcelain tile or a VDRL test plate, pricking device such as a lancet or pricking pen, alcohol swab or a cotton moistened with rubbing alcohol, toothpicks, matchsticks, or capillary tubes. ABO blood grouping test kit consisting of antisera A, B, and D antibody vials. To begin the test, first remove the ABO blood grouping test kit from the refrigerator. Homogenize the contents of the antisera antibody vials by inverting each bottle 3 to 5 times and allow them to equilibrate to room temperature. Meanwhile, take the pre-cleaned glass slide and place it on a white sheet of paper or porcelain tile. Next, surface sterilize the tip of the middle or ring finger using an alcohol swab. Allow the alcohol to dry and then gently but briskly prick the finger using the lancet or the pricking pen. Always remember to use a new lancet or pricking needle for each person. You may or may not wipe off the first blood ooze. Now gently place 4 drops of blood in the glass slide at more or less equal distances from each other. Do not place the 2 corner drops too close to the edges of the slide, as it might cause some inconvenience when mixing later with the mixing stick. Then open the vials and place 1 drop each of the antisera A, antisera B, antisera D reagents, in quick succession to the first, second, and third drops of blood respectively. To the 4th blood drop, which is the negative control, add nothing else but a drop of normal saline. During reagent addition, always take extra care not to touch the tips of the reagent droppers with any of the blood samples. Once the 3 antisera and normal saline have been added to the test and control blood samples respectively, quickly and thoroughly mix the 4 blood drops individually using 4 different toothpicks or matchsticks. Do not and never use the same mixing stick to mix all 4 drops. Once you have individually mixed all the reagent blood drops, leave the slide undisturbed for 30 seconds to a couple of minutes. At the end of this time period, check for agglutination or clumping of RBCs in the 4 mixture drops. For quick reference, clumping or agglutination looks something like the photo on the left as compared to the photo on the right, which shows no agglutination. The negative control is not expected to show clumping, in which case the experiment needs to be repeated. The blood group of the tested blood sample will now depend on which of the 3 drops on the slide exhibit RBC clumping. This is a